Hello everyone, I'm Habiba Sweetie. The name of our group is Group Flower and the topic of our presentation is sample preparation for volatile organic compound. First, we have to know what is volatile organic compound. Volatile organic compound are a group of organic chemicals that can easily vaporize into the air due to their relatively low boiling point. They are called volatile because of their ability to vaporize readily. Um, they are often found in various environmental matrices, including air, water, soil, and biological samples. Volatile organic compound can be a concern when they are found in surface water for several reasons. First one is environmental impact. Volatile organic compound can have a harmful effect on aquatic ecosystem when this compound enter surface water. They can contaminate the water and impact the health of aquatic plants, animals, and organisms. Some volatile organic compound can be toxic to aquatic life, disruptive food chain, and ecosystem balance. Second one is human health. If surface water contaminated with volatile organic compound is used for drinking water or recreational activities such as swimming, it can pose risks to human health. Some volatile organic compounds are known to be carcinogen or can cause other health problems when ingested or absorbed through the skin. And last one is order and taste. When even at low concentration, some volatile organic compound can impact unpleasant odor and taste it to drinking water and making it unpalatable. This can lead to decreased water quality, public dissatisfaction with water supplies. And why does the sample preparation is important? Sample preparation for volatile organic compound in some surface water is essential for several reasons, such as detection sensitivity. Volatile organic compound are often present in trace concentration in surface water. Proper sample preparation ensures that this low level can be detected and quantified accurately by analyzing instruments such as gas chromatography, spectrometry, or liquid chromatography or mass spectrometry. The contamination control. Surface water sample can easily become contaminated during collection, handling, and storage. Sample preparation techniques such as filtration, preservation help minimize the risks of contamination and ensure that the measured volatile organic compound concentration are representative of the actual water sample. Um, then preservation of sample integrity. Volatile organic compound can be highly volatile and degrade over time. Proper sample preparation involves the use of proper preservation methods such as acidification or refrigeration to prevent the loss or transformation of volatile organic compound before analysis. And say another one is removal of interfering substance. Surface water samples may contain various non-target compound, particular matter, or suspended solid that can interfere with the analysis of volatile organic compound. Sample preparation method like liquid extraction and solid phase extraction can help isolate and concentrate the target volatile organic compound and reducing interference. Thank you.
The Rio Grande River in the United States, known as the Rio Bravo, also in Mexico, is a river of 1,885 miles and is the fourth longest river system in the United States. Also, the Rio Grande marks the boundary between Mexico and United States. Also, the Rio Grande irrigates the farmlands in the middle Rio Grande Valley to the desert cities of Albuquerque and Las Cruces in New Mexico, and also to El Paso, Texas, and then to Ciudad Juarez, Chihuahua. The presence of cities, urban areas, and also agricultural sites has an impact on the river health. Also, it's important to say that the International Water Treaty also have an effect to the river's health. You can see how it has changed the river across the time, over the time. There are more cities around the river, there are more farmlands, there are more agricultural sites, and there is more impact to the river. And also, maybe this impact in the river are affecting the human health, and also probably, very probably, are affecting the environment around the river. The goal of the project is to evaluate the presence and concentration of volatile organic compounds in surface water samples. The timeline is going to be in the Elephant Butte Dam release from June to August. And also, the lot is the Rio Grande River from the county of Sierra, New Mexico, Doña Ana, New Mexico, and also El Paso, Texas. For this project, we determined 7 sampling sites across from starting from the Elephant Dam across Old Sierra, Doña Ana, and El Paso, Texas, to the border with Mexico. It's important that we take samplings in urban areas, also in agricultural sites, and also in dams, and also in water bodies across the river. So we determine these sampling sites using these variables. Also, these sampling designs seeks to have a georeference diagnosis to identify sources and the possible relationship of urban or agricultural areas with the presence of VOCs. This is one example of the sampling location. This is the first sampling and is between the Elephant Dam and True or Consequences cities. The second, in this case, the second sampling site is after the city of True or Consequences. This can help us to know if is a variability between the sampling in the before the city and the sampling after the city.
All right, now to discuss our sampling plan as well as sample matrix. So our samples will be collected at regular intervals throughout the course of our sampling period. We are going to be collecting our samples bi-monthly. Our sample matrix is going to be the grab water samples that we are going to take from random points along sampling areas in glass bottles. During transport, we are going to keep these samples chill to avoid any sort of tampering. When we do get those samples to the lab, we are going to remove impurities as well as aliquot representative samples into smaller volumes. All right, now to discuss our analytes. So these are the organic compounds that we are specifically targeting for analysis within our samples that we have collected at our sample site. So to do this, we are going to be using the purge and trap method. However, I will define the analytes that we are particularly interested in. This can include benzene, which has carcinogenic effects, toluene, which can cause pneumonitis, and these are also examples of BTEX compounds. Typically, these are released from emissions from motor vehicles as well as aircrafts. You can see in this table that we have listed the compound as well as the response and the stability. So both of these compounds are highly stable in preserved water samples for greater than 60 days, which is great for the way that we have preserved our samples.
Hello, everyone. This is Arturo Rocha, and what I'm going to do is I'm going to go ahead and explain these volatile organic compounds again uh, from our study, and surface water, of course, uh, referring to the Rio Grande. So this these slides are going to primarily describe EPA method 5030C, which is also known as a purge and trap method for aqueous samples. So this method is designed for the monitoring of, again, these VOCs in water. And again, some of these compounds include those BTEX compounds, which are those aromatic compounds, which include, for example, benzene and toluene. And it allows a large amounts of methanol, which is going to be the solvent, to pass through, in this case, the absorbent beds and attaining the analytes of interest. So one of the first things we need to do is, again, from obtaining the samples from the river, we need to filter out any other media that could interfere with the results and also cause, in this case, any other kind of reading, for example, like false positives or false negatives. So, provided below is kind of like a flow chart of, it's kind of like a dichotomous key flow chart of uh, the actual purge and trap method, of whether it was a successful or not a successful way to, in this case, do the test. So we begin with our initial water sample, and of course, usually as a we insert the water sample and in conjuncture with gas. So, for example, it could be helium, and of course, it will be combined with methanol, and it will, in this case, be introduced, in which, of course, it needs to be actually controlled by temperature. So, a relatively high temperature of about 80 degrees Celsius, and then from there, it will, of course, the analytes of interest and the solution will change its actual physical state from a aqueous solution to a vapor phase. And this is the part in which you can have two results. So if it's a positive result, they're gonna usually shown here by the arrow that says yes. So this is the purge and trap method. So it will, in this case, try to retain the analytes of interest. And then the trap is then heated and then backflushed with the inert gas to desorb the purgables. And then finally, it will be sent to the gas chromatograph column, which is the GC method. Now, if something goes wrong within, in this case, that initial state here in the orange diamond, in which you get a negative result, that usually due to contamination samples. You might have a high concentration of analytes, and in which you have to do several steps. You might want to check for cross contamination, and also any kind of organics that, in this case, are volatile. You might have to, in this case, have a round of diffusion of which, so you can, in this case, might have to prepare some trip blanks. So trip blanks are pretty much are samples in which they're free of analytes, in which they contain a media. In this case, it will be an aqueous solution, and they're collected in the same type of container for the test. And then once that is done, then you can go ahead and uh, possibly use it back for the actual purge and trap method. Okay, so that was the pre-preparation, the purging gas, in which if it passes through a positive result, it is then sent to the analytical methods. That's going to be the GCMS, the gas chromatography and mass spectrometry. So there's various actual methods for this, and usually they begin in the 8000 series of the EPA methods. Now, I have wrote here 8260, and the one that contains some compounds, especially those benzenes and those related isomers, those BTEX compounds will usually rely on EPA method 8021. Okay, and that is our sources for our presentation and project. Thank you for your time.